Thank you. I'm very happy to have an opportunity here to address a local group. I'm just down the road in Kenilworth, New Jersey, and great to see the stimulating conversation about microbiome here in New Jersey. I do want to say that Merck has not had necessarily a focused effort on microbiome research. I'll tell you a little bit about what we are doing, and these are really my views, given all the different therapy areas represented by Merck and a lot of different approaches to how microbiome research may be used. Just make it clear this isn't necessarily the comprehensive view across the organization. So really currently within Merck, because we are a major antibiotic company, a company that sells primarily antibiotics in the hospital market for severe infections, and we've just expanded that tremendously with the acquisition of a company, Cubis, that was up in Boston. We are leaders in advocacy for antimicrobial stewardship as a recognition of making sure antibiotics are being used appropriately, not just to prevent resistance, but with this strong recognition of the role of the microbiome in health. We want to make sure that antibiotics aren't being used inappropriately where they may cause more harm than good. The other aspect is we do have now C. difficile therapies, and what we're really trying to further our understanding of is how these drugs affect the microbiome. And we know that the microbiome is a critical part of C. difficile, both the cause and the therapy. So we have both, again, through the acquisition of Cubis, the first narrow spectrum agent, Difficid, that was developed specifically with the idea that because of its narrow spectrum of activity, it would reduce the opportunity for recurrence of C. difficile disease. And we have a new entity, which is still under the review by the FDA, although we hope to hear shortly of their decision on this product, which is not an antibiotic, but monoclonal antibodies that are directed to neutralize toxin B. Now, in addition, because of the recent recognition of really the critical impact of microbiome and human immunobiology across a huge area of disease types, as I said, we have not had a really integrated approach to how we thought about the microbiome, and we are opening a brand new exploratory science center in Cambridge, Massachusetts, and the center will be focusing really on human immunobiology, where we're taking a very disease-agnostic approach, looking at host pathogen and host commensal or microbiome types of interactions, and trying to better understand how we could leverage that to improve therapies. Merck is a leader in the checkpoint inhibitors with the PD-1 compounds, but we know that a small subset of patients still do respond to those therapies, and trying to understand where you have really perfect immune homeostasis, I think, when you have the gut microbiome interactions with the host, where you're controlling disease and invasion while also maintaining beneficial commensal organisms, is really kind of an ideal situation, because we know the sterile gut is not the healthy gut. It's really that very fine balance that's maintained through those interactions, which are critical. And I think that will help us understand more how we can control and utilize the immune response in a number of disease states. And this is just my chance to shamelessly plug the fact that this center is just opening. We have a number of positions posted at the Boston site. If anybody's interested in multiple views of microbiome research, please look this up on the Merck website, and look forward to finding new innovative scientists. This is being set up actually as a subsidiary organization. It's going to be tried outside of the Merck bureaucracy, we might say. So it's supposed to be very nimble and very interactive with the local communities. So it's kind of an experiment within itself as to how we're trying to make this work, and so I think it's going to be very, very exciting. Now I'm going to focus a little bit on C. difficile therapy, which we heard a little bit about already from Dr. Honig. I call this a therapeutic example, which is supposed to be a bit of a double entendre, because we've talked a lot about manipulating the microbiome and the role of microbiome in disease, but we have these concerns about causation as opposed to correlation. And this is clearly a case where we know when the microbiome is disrupted, these patients are potentiated for disease. And that dysbiosis then leads to the opportunity for infection with C. diff spores and growth. And we do know that microbiome therapies, particularly the fecal transplant therapies, have been widely successful and recognized as a way to prevent recurrence of disease. And as we've heard, though, there's tried to be a progression of how we can take that knowledge about fecal microbiota transplant and turn that into products such as pills, which may be fecal pills, which are controlled, or eventually into these defined organism groups that will have a similar effect. But as we also heard from Dr. Honig, as we start to get too defined again with this therapy, as Series has seen, where they've really tried to reduce that down to specific organism groups. The results in the clinical studies were not positive, and so it clearly shows there's still a lot to what we're deriving from fecal transplant therapy that we may not fully understand yet, and a better understanding of those interactions with those organisms is going to be critical. Now I'm going to tell you our experience about the systemic C. diff antitoxin antibody approach, which is another method to prevent recurrence of disease, and I'll tell you a few things about this because I think it's a bit of a cautionary tale on still what we think about with C. difficile, where this is probably what we think about as the most simple and direct example of microbiome therapy and how we're utilizing it. You'll see that we really confronted a lot of surprises when we went into these studies. So the phase 3 study I need to go through it a little bit because it's been raised as a bit of an issue in the way that you even look at and study and understand the impact on recurrence of C. difficile disease. The way that we tried to design these phase 3 studies was to really replicate the way we felt that physicians would be wanting to use this therapy, and that is when they are treating a current episode of C. difficile. During that treatment, they're allowed to then provide the antibody infusion. It's a single antibody infusion against the toxin B. And because of that, there's this concern that it's being given during therapy, and therefore, when you actually assess efficacy, you should be assessing both the cure of the initial episode, and then really what we wanted to assess was the long-term endpoint, which we studied for 80 days post the cure of the C. diff episode, of whether or not there was recurrence of disease in the group that got the antibody versus a group that was treated with standard of care antibiotics and didn't get the antibody therapy. But the challenge that we've had in the statistical analyses of these data was this component of, well, first you have to cure the disease before you then can assess recurrence, and if you don't cure the disease, how do you then calculate recurrence as the endpoint piece of that? And that's where it's been a little bit challenging. And the FDA's favorite approach to taking this, at least with the current narrow-spectrum antibiotics, has been its global cure, which is curing the initial episode and preventing recurrence, and so it's a bit challenging. A couple other things I wanted to point out, which I think is important here, is during that 80-day episode where we were looking for recurrence of disease, probably at least a quarter to a third of those patients required additional antibiotic therapy for other infections, not because of C. difficile infections, but they needed antibiotic therapy because these are typically very sick patient populations within the hospital that are highly susceptible to disease. So if you had had a microbiome therapy or another therapy, probably then that wouldn't have been effective once you had to go back and have the antibiotics to treat other diseases. And I also want to point out these antibodies are fully human antibodies, so they have a very long half-life, so we know we actually have neutralizing levels of these antibodies in circulation throughout that entire 80-day period. Now, the other question that comes back, these are systemic antibodies. They're circulating in the bloodstream, but what we've observed in our hypotheses of how these work is that when you get the C. diff toxins beginning to damage the intestinal lining, you immediately have a diffusion into that place of damage, and the antibodies are there then to neutralize and reduce further damage and not lead to then the more extreme symptoms of the disease. Now, this is the results from the two different studies and then the combined data. And what you'll see in the Modify 1, which was the first study, is this is where the FDA required us to do a study to address what's called the combination rule. Because the fact is, all the biology that we knew about C. difficile was that C. difficile makes two toxins, toxin A and toxin B. All the data that we had from all the rodent models said you have to neutralize both of those toxins in order to have an effective therapy. We really wanted to go in and do a two-arm study, combined antitoxin A, antitoxin B, versus placebo. Well, the FDA said, well, nobody's ever done a definitive experience to say you need both of those antibodies. Really, do you know that you need both of those? And so in that first study, we had 4 groups. One was just antitoxin A, the other was antitoxin B, the other was the combination, which we anticipated to be our product, and then we compared that to placebo. Well, we had an opportunity to do an interim analysis to see whether or not there was going to be any futility in the single monoclonal antibody arms, which is kind of what our assumption was. But in fact, when you got the final data set, what we saw was antitoxin B by itself had just as good of efficacy as the combination of antitoxin A and antitoxin B. And antitoxin A had no better efficacy than placebo. So in the human disease, the data, the clinical data really suggests that toxin B is really the only toxin that was driving these aspects of recurrence that we were measuring in the C. difficile disease. And so you can see that then when looking at this combined data, by using this therapy, so adding this antibody on board during the treatment of an episode of C. difficile with standard of care antibiotics, you have a reduction of 10% in the overall recurrence rate then in the severe population, which when you do the relative risk reduction, it's about a 40% relative risk reduction in recurrent C. difficile based on these antibody therapies. Now, I kind of gave away this punchline already, but as I said, when we went into these studies, essentially, we had, the hamster model, which has always been the gold standard model. And when you look at that hamster model, in fact, toxin A did look to be more important, and there was a lot of discussion in the literature about really what was more important, toxin A or toxin B. And what we saw was antitoxin A had marginal efficacy in the hamster. B had nothing, and when you combine the antitoxin A, antitoxin Bs, in fact, you had full efficacy. In mice, we saw a bit of a mix when we've done these models. And again, the combination was absolutely required to have full efficacy. Now, something that we had done with a group in Tufts was a gnotobiotic pig model. And in that pig model, they used both our monoclonal antibodies and polyclonal antibodies directed against antitoxin A and antitoxin B. And they actually replicated the human result. What they saw in the piglet was that antitoxin A had no efficacy. Antitoxin B provided all of the efficacy with no additional efficacy associated with neutralizing toxin A in that group. So in this case, this is an instance where the gold standard model that we've all used to be thinking about this disease was probably the pig model and not the hamster model, which is what we've always talked about. So that's kind of one cautionary tale about how much do we really know still about some of our rodent models for some aspects of microbiome-related diseases. Another analysis that we were looking at was still mechanistically, question is, so how does neutralizing toxin prevent recurrence of disease? So we know the toxins are required for the symptomology of the disease, so how does neutralizing that then lead to the lack of recurrence? This is just looking at the general data on survival when you've treated with either vehicle, where you see in the hamster model, it's a very severe model, the hamsters die almost immediately in that group. If you treat with vancomycin, which is kind of your antibiotic standard in that case, you see that you have 100% survival out to 10 days, but 10 days was then when we stopped the antibiotic therapy, and once you stop the antibiotic therapy, you get a rebloom of those C. difficile spores, and then you see almost 100% lethality. Whereas when we've prophylactically treated with the monoclonal antibody, so this is before the C. difficile infection, but it's just a single dose, you see that you have survival for 100% survival for 5 days. Then you start to lose some of the rodents, but then you still have about 50% survival. In the mice, you see a very similar effect. The mouse model has never been as severe as something like the golden hamster model. But here again, with vancomycin, you see 100% survival until you stop the vancomycin therapy, and then you see a decrease down to only about 20% survival. Whereas here again, with the prophylactic antitoxin antibodies, you see almost 90% survival just provided by the antibodies. Now, within that study, we said, well, what's going on with the microbiome in those mice that have gone through these therapies? And if you look at the vehicle-treated group, which is in the upper left, the solid points on the left are actually C. difficile spores that are measured in the feces of those animals. So after you do the induction with the C. difficile spores, you see a high spore count. You've ablated the host immune system, but what you see with the principal component analysis with the axis on the, excuse me, the y-axis on the right side, is that microbiome quickly rebounds after you've stopped the antibiotic ablation therapy before you treat it with the C. diff spores, and you see really a perfect correlation of the recovery of the microbiome and the decrease in the prevalence of C. diff spores in the feces. Now, if you look at the vancomycin group, in the lower right-hand corner, what you see is that the vancomycin actually suppresses the C. diff spores, so you don't see that big early spike in the C. diff spores. But what you do see, once again, is that on day 10, when you've stopped the antibiotic, all of a sudden the C. diff spore count goes extremely high. And you can also see that when you look at the recovery of the innate microbiome based on the principal component analysis, is it doesn't start to recover until you've stopped that vancomycin therapy. So what you've done with the vancomycin is suppress the C. diff, but you've also had an impact on the microbiome. And so once again, the C. diff spores have tended to overgrow, and that's why you end up with the high lethality then with the vancomycin treatment, because you haven't eradicated the C. diff and you've suppressed the microbiome. But then if you look at the actobezlo treatment group on the lower left corner, it looks very much like the vehicle control group, where you essentially have an initial burst in the C. diff spore, so you really haven't affected the C. diff infection at all in those organisms, but you also haven't impacted the recovery of the microbiome. And because you're getting that rapid recovery of the microbiome without the antibiotic use, that's what gives you then the broad protection within that model. So when you think about recurrence, clinical recurrence of C. diff in the hospital, the issue is this recurrence cycle where you're using antibiotics constantly to treat the actual current episode of C. difficile. That leads to this disruption of the biome, which I'm showing in the blue line, and so then allows recurrence by overgrowth of C. difficile spores. So what we're doing with bezilotoximab, so we're adding it during the treatment of the first episode of C. difficile. And because now we have an antibody in circulation, which neutralizes the effect of the toxin, is that even though you probably still have a C. diff bloom once you've stopped your antibiotic therapy, the antitoxin antibodies are neutralizing the effect of that toxin, so you don't have symptomology of C. difficile in that patient population. And because there's no symptoms of diarrhea and disease, the doctors aren't thinking that I need to treat this with antibiotics. And because you don't treat it with antibiotics, you actually then allow that microbiome to recover within that patient while you've protected them from the symptoms of the disease with the monoclonal antibody. And then that's what we feel leads to the actual lasting effect on prevention of recurrence within that patient population. Which is why we think that while this is not a microbiome-directed therapy, it's in fact a therapy that we think is completely dependent on the microbiome to help prevent recurrence of disease in these patients. And so those were just a couple tales around C. diff to kind of explain, again, as I said, what people think is a very simplistic model of the role of the microbiome, but there's still, in the way that we model this and understanding the role of the microbiome in these diseases, is still quite a bit of complexity.
Thank you, Todd. Questions?
Did you have any issues with immunogenicity of either monoclonal?
No, we haven't. So another examples come back. So we've done those multi-dose studies in healthy volunteers, and so we've have not seen any immunoreactivity. We haven't seen any anti-drug antibodies being generated at this point. We haven't, as a criticism, we have not done those studies in patient populations, and so whether or not there might be a difference there, we don't know. We know people have endogenous circulating antibodies to these toxins, and in fact, maybe one of the risk factors is people who have low responsiveness and low natural titers are the ones who have the greatest benefit from the product, in fact.
Thank you for the fascinating presentation, and especially thanks to Merck for being so forthcoming and sharing in such great detail your clinical experience with this really innovative approach. I was wondering if you could just comment generally from Merck's perspective and in general from the pharma perspective about this general issue of attacking infectious diseases and other pathologies by using strategies that are effectively modulating the microbiome or modulating the effect of other interventions on the microbiome in a host pathogen setting, and whether that there are opportunities for drug discovery and other fields moving forward, and whether that those kind of arguments, as your group presented at the FDA at the advisory council meeting, this very powerful argument about the mechanism, whether that will eventually be translated into more formal guidelines from regulatory agencies about how our medications that we're developing are affecting the microbiome and human health in general.
Well, to answer the last part first, I've learned there's no way I can predict what the regulatory agencies are going to come up with in their analyses. So one of the aspects I didn't point out with this center, the exploratory center that we're forming up in Boston, is Merck is not just a drug company, but we're also a major vaccine company, and finding ways to improve, especially adult vaccine responses, is another key component of what we'd like to do, of understanding how you might have better adjuvant components, and it may be associated with, through stimulation of the microbiome, or understanding, what the role of that, gut health may have in terms of people's responsiveness to vaccine antigens. So yeah, to try to get away from treatment, but actually having prevention components are certainly a critical part of what Merck is engaged in. And I think we've all been completely awed by the checkpoint inhibitor responses that we've seen in oncology, and I think in infectious diseases we've been trying for a while to find ways that we can modulate response to infections. And we know it's challenging to find that fine balance of overstimulating versus understimulating, where either you're at risk of infection or you're at risk of sepsis. And so, it does give us some hope that you can maybe are starting to find some entries into that, where what we're learning in oncology, maybe we'll eventually be able to translate that back into taking non-antibiotic therapies for infectious diseases also. And of course, in our HIVs, we have a lot of HIVs, so some of these components too could fit into some of our efforts to cure HIV. Are there ways that you can, try to stimulate the reservoirs and eradicate, and actually have an eradication strategy, which I think will also be impacted by some of these types of studies.
If I understand correctly, your antibody is almost acting like a self-fecal transplant, because you're wiping out the C. diff, and you're not wiping out the bacteria in the gut with antibiotics, so you're allowing them to regrow. Is that how you see it?
So we're not wiping out the C. diff, so we have no effect on the C. diff, but
Well,
what we're doing is
not that you're wiping out the toxin.
Yes,
so
we're
you're not
preventing-
giving it any selective advantage because you haven't treated with antibiotics to give it a nice space to live.
Yes, so we think we're just preventing that symptomology, and without that symptomology, there's no need to treat with the antibiotics to eradicate the C. diff.
I just thought of another question. Does the toxin have any direct effect on the gut microbiome?
So that was one of the things we were interested in, that microbiome study that we performed, and so we did not see any difference in that principal component analysis that people who received the antitoxin therapy had any difference in that native microbiome that recovered, because that was one of the key questions, in fact, that we wanted to understand. Because again, is toxin, what is the ecology of the toxin? Why is it there to help, is it really there to help, maintain? And no, we did not see anything where there was a direct effect on the microbiome with the toxin or with the neutralization of the toxins. That was actually a full metagenome analysis that we did. So that was every organism, that principal component analysis we did in the fecal samples, but that was.
I'm going to ask Christine to join you again for a minute. A few more minutes.
Yeah, so Todd, I just had a quick question in regards to the new lab that you guys are opening up in Cambridge. Is that focused I wasn't able to catch it, but are you guys focused particularly on microbiome itself, or is this is it focusing more on microbiome research, like discovery research, or is it just more along particular disease areas?
Well, like I said, I think the focus is kind of human immunobiology, and so it's really, what do we know the most about in terms of some of this human immunobiology, and it is host pathogen interactions, or with the microbiome, I consider that kind of host commensal types of interactions. So my boss, Daria Hazuda, has been named as the chief scientific officer. As I said, it's being set up as kind of a subsidiary, so it's a little bit of an unusual organization. And while it's not infectious disease focused, it's really trying to look at these understandings of interactions between commensals or pathogens and the host and those responses that we think will impact not just infectious disease but many disease states. So it's microbiome is just a subset of that. It's not the absolute focus of the institute.
Todd, I just had one other question. So is there are there any other therapies, antitoxin antibodies for like foodborne toxins or anything like that? I'm not aware if there are or not.
I think the only other toxin I know of is the anthrax, where there's monoclonal antibodies to anthrax toxin. I think that's the only other antitoxin. There's been a lot of discussions about anti-Shigella toxins and diarrheal toxins. The problem is those are more acute diseases, and by the time you know you got the disease, it's hard to necessarily intervene, and the recurrence component isn't as significant in that. So it's hard to see that there's necessarily a clear market for how you would utilize that.
There's a theme that's come up a number of times that I wonder if each of you can comment on, and that's about the challenge of developing commercial products from such a complex and variable environment. Howard made the point with yogurts about whether the lactobacillus is the same, directly applicable, obviously, to your products, Christine. The point was made by Gerard about fecal transplantation. The exact composition of microbiome varies with time and diet. It varies in the same individual. If you limit sampling to fecal samples, are you missing what's happening in the higher regions of the gut? And then finally, the complexity to manipulate the microbiome beyond a single or small group of bacteria. I wonder if you might comment on how that set of questions influences both of your approaches and perspectives on looking at the microbiome as opportunities for therapeutics.
So as far as the first question goes, I kind of highlighted that in my talk, but just to kind of reiterate it and go into a little bit more complexity. So as far as human milk oligosaccharides go, there is a vast amount of different actual human milk oligosaccharides. And so obviously that makes it very complex when you have different women producing different ones and at different amounts and at different times. And then when you have the added complexity of actually making these things, because hopefully not surprisingly to the audience, people don't actually want to give other ladies human milk oligosaccharides to their own babies. So we have to make them ourselves. And so the thing is that, cost becomes a huge issue, and then which one? So a lot of the work that we have done is to try and kind of tease out which one will provide the most benefit for the least cost. And that's kind of how we, when we look at, say, fibers like inulin or GOS or FOS. We look at those types of issues as well. And even in some of our other, like our proteins and fats, you would be surprised how sometimes this stuff can be extremely pricey or relatively inexpensive. But it's definitely a cost-benefit analysis that we do. And I guess to your other question about looking at different kind of places or whatever to kind of, obviously the whole baby, the whole adult, the whole child is filled with a variety of different organisms. And, no one scientist, no one company can do it all. And so you kind of pick and choose, I think, what works for your group, your goals and initiatives. And sometimes you end up finding a lot of really cool stuff. One of the things we found with human milk oligosaccharides is that, okay, obviously we know it has effects on the gut. That's not a big surprise. But if anybody here has ever watched an infant feed, especially when they're breastfed, they get human milk in their nose, in their eyes, in their ears, everywhere. And what we found is that human milk oligosaccharides actually have beneficial effects for reducing otitis media. So just because something maybe has an effect on the gut doesn't necessarily mean that it doesn't have an effect other place. So I think kind of, really looking at things closely and kind of what Merck's doing with C. diff. And, you may think that you know all the answers, but after a few years of research, you may find out that you're just kind of discovering what the questions are.
I think that's probably the key phrase. We're just discovering what the questions are. I think what Dr. Boston said this morning, as a microbiologist, when we started to see this information on the impact of the microbiome in human health, it was extremely exciting. But I guess also as a microbiologist, I thought, duh, we've known this, but, in a sense, I don't think what we've, were able to do was ever start to analyze that. And so I kind of think about these metagenomes as, I like to call it the dark matter. Essentially, this is something that we've just sort of discovered. There's so much that we don't know about what these interactions are. I think the work that we're talking about doing in the CTM right now is just absolutely critical, because people are using multiple methods to really even understand, or define what the microbiome is. But recent studies have shown that you see more diversity by using a different method on the same sample than you see by doing a study across samples. And so there's so many of these basic questions that I think we're going to have to get to before we can really start thinking about using this on more of a therapeutic modality, understanding it. I think the series experiment was a bit of that too. I think it was a very simplistic approach. Okay, I'm going to take all these, sporulating species and put them into a patient, and I'm going to see a therapeutic effect. And I don't think, again, when you consider what I showed you about how translatable are the models, how much do we really know about what those models are telling us, and to take that right into a clinical study without having some validation of what that product is, I think is where the challenge is. Anybody who's worked on biologics, the whole question of bioequivalence of even a monoclonal antibody. Somebody's trying to make a monoclonal antibody just to replicate somebody else's, and we've had this big challenge of, well, how do you say this is bioequivalent, even though it's binding the same antigen, it's the same PK profile. So when you've got something where, especially if you're taking it from, a sourced human sample, how do you qualify that as being biologically equivalent? So I think there's a lot of key questions about how we're going to be able to turn these into products, and some of the basic science even, to really understand more of how the biome is interacting with us, and how we might utilize that.
Thank you both. Any other questions?
I have one more question about the 2FL. In your study, how did you select infants for that study? Did people volunteer to have
random.
But did people volunteer to have their infants take the formula with 2FL? Or?
Yes.
Uh-huh.
Yeah. Exactly.
So you're doing randomized trials. I mean.
Yes. Yeah, and the thing is, that's how we kind of were able to know that 2FL is produced in about 80% of women. Granted, we're not doing studies in other parts of the globe. However, our infant products do go to different parts of the globe. So in the future, that's probably the direction we're going to go when we expand this product globally. So of course, then we'll find out more information. But yeah, it's very difficult to enroll babies in trials. And so obviously, making sure that the ingredient is safe first and foremost before we even test it. So I know there was a question about kind of animal models and difficulty. Again, not surprisingly, it's really hard to get mice and rats and hamsters to have human milk. So we have an extremely difficult time with animal models. However, that doesn't mean that we don't use them, because you can't just put something into an infant formula and say, hey kids, let's try this out. But yeah, we also do a lot of analysis of breast milk, and we are getting samples of breast milk is relatively easy. So we do a lot of analysis, find out what women's profiles are, and we do a lot of work with the mother-infant dyad. So we tend to not piece them out separately, because so much of what happens in mom is happening in infant. I didn't mention, but HMOs actually have been shown to have positive effects on breastfeeding women too. They actually have been shown to reduce incidence of mastitis. So women that don't produce 2FL specifically tend to have higher incidences of mastitis, which unpleasant.
Have you ever thought about making a diagnostic so that women could test their breast milk and see what HMOs
If I worked
has?
in Abbott Diagnostics, I'd be all over it right now. Yeah, Abbott is a huge organization, if you're not familiar. 2012, we divested our brand pharmaceuticals here in the U.S., but we still have a generic ex-U.S. pharmaceutical company based in Basel, Switzerland. but we do have a diagnostics division, and some of the work I do with connecting with researchers and institutions. Recently, I was at Stanford talking about microbiome work, and we did actually bring in our diagnostic colleagues just because they have expertise and in that area. So we want to make sure that if they hear anything that sounds cool, like, let's start talking about it. Let's see if we can build this into a bedside test. So yeah, we're definitely thinking about it, just not me personally.
Well, thank you again. Howard, I'll pass to you.
Okay, thank you. So thank you very much. And so I'll just try to sum up the meeting today. I only have 47 slides, so we should be out of here in just a few minutes. But no, I think it's really was a very, very interesting meeting and very different from many of the meetings I've been to, because we had a full range of gamut of discussion from basic science to products in the field that are going to change and improve human health. I had no idea of the National Academy study on the buildings. I think that could probably last 100 years, given it's a huge undertaking that Dr. Bennett told us about. Then we heard how the microbiome in infants may infect them for their whole life and may be inducing inflammation and creating problems for them, especially the premature infants. And then we heard Laurel how they're trying to really make products that can change the vaginal microbiome and improve women's health. We heard two great talks, one by Romina about the effects of the microbiome on cancer therapy, and that's becoming more and more a big deal as people are beginning to try to identify organisms that might benefit or that patients should have before they undergo immunotherapy. And then Dr. Manny told us about a real basic research but fascinating story about swarming bacteria. I had never thought about this. I think he's really thinking outside the box, and it could be a new approach to reducing gut inflammation. Then in this afternoon, we had two great talks about the need for standardization. I think that's an overriding area that everybody realizes has to be done, and it's difficult because it's not something that you're going to get into science. I mean, it means people are going to have to work at this and publish it where people can read it, but they're not, like I said, it's not going to be a high-profile journal because they want something a little different. But it's still absolutely necessary if this field is to advance properly and really make a difference in how people are diagnosed and treated for disease. Then we heard a really interesting talk about ancient DNA and how that's going to tell us a lot about evolution and how people's diets changed and their microbes changed. And the tools are becoming better and better for that. And finally, this afternoon, we heard from two companies that their products are really, the microbiome impacts their products, and they're trying to develop products that can change the microbiome, but not use microorganisms directly. So it's really, like I said, been a very unusual and rewarding meeting, and I hope you all felt that the 6 to 7 hours you spent here was worth your time, because it really gave different perspectives on different aspects of the microbiome. So I want to thank all the speakers and all the participants for being here and really enlightening us into new areas that are forthcoming and will be continually expanding in the future. So thank you.